Good evening, good afternoon, and welcome to the ASPN webinar this evening. Can we change the practice of SCS with biomarker-based dosing? This is a very exciting webinar. We are thrilled you're joining us. The webinar will start in just 1 minute to allow people time to log on. Again, if you're just now logging in, welcome to the ASPN webinar this evening. Can we change the practice of SCS with biomarker-based dosing? The webinar will start in just a moment. Good evening, everyone. If you are just now joining us, welcome to the ASPN webinar. Can we change the practice of SCS with biomarker-based dosing? We are thrilled you could join us this evening. The webinar will start in just 1 more minute to allow everyone the opportunity to log on. Again, welcome to the webinar. We will start in just 1 more minute. Good afternoon, good evening, and welcome everyone to the ASPN webinar. Can we change the practice of SCS with biomarker-based dosing? Drs. Deer and Syed, take it away.
Thank you, Michelle, for the introduction, and thank you everyone for logging in tonight. We are today, we welcome you to join us on what I think is, a really important topic and question we're going to tackle. Can biomarker-based outcomes reshape the field of spinal cord stimulation? I've often kind of spoken with my colleagues in cardiology and endocrinology, and they also work with medical devices, and I've always been really jealous at the way that they can see and assess how their devices are working. They have biomarkers. They can tell. It's not really what the patient says, it's the patient in a bad mood. We have a very different thing that we've always kind of dealt with in the world of chronic pain and spinal cord stimulation for that indication, where it's very objective. So we're going to tackle that question. Can we apply biomarker-based strategies to optimize our therapy so we can really assess how our patients are doing? Tim is really kind of killing us all in his background today. He's live from New Zealand training for another ultra race, a triathlon, I believe. So Tim, this is our crew here: Dr. Deer, Dr. Anthony, Dr. Peterson, and Dr. Pope. We'll go to the next slide, please. Click in there a little bit, but as we're waiting for that, we'll just speak a little bit about kind of today's program. So again, all of the faculty today are functioning as paid consultants for Saluda Medical. The opinions that are expressed today of the speakers are really not necessarily those of Saluda Medical, and they're not really necessarily the opinions of the ASPN Society. What we're trying to do here is give medical education on breakthrough technology for our members in a way that we feel is very ethical and evidence-based. So Tim, what do we hope to learn from today's webinar? Welcome.
Well, thanks, Dawa. Great introduction. I think you're right. When I do something like basivertebral nerve ablation, I see those implant changes. There's a biomarker there that helps me predict the candidate. We've never really had that with spinal cord stimulation. The question now is that a possibility? We're going to talk with Dr. Pope initially about the landmark Evoke study, which many of us had a really honor to be part of, which was with the smart loop technology that he's going to talk about. And then we're going to talk about really, with Dr. Anthony, can we make differences there by individualizing that therapy based on things like the therapeutic window and the ECAP response. And then we're going to have Dr. Peterson conclude with really objective biomarker-based dosing and really bring that theorem that there is a possibility of biomarker-based dosing to the world. And so I think that's going to be very exciting, the whole discussion, Dawood. I think we'll learn a lot. And our first speaker, a dear friend of mine for now quite some time, who I think does a lot of the world's leading research in the field, Dr. Jason Pope. Jason, the podium's yours, my friend.
Hello, team. Well, thanks for the kind introduction. And I was chatting with Tim earlier. He is obviously lakeside, and I am in my practice. So one of us is living our best life. I haven't decided which. And I appreciate the opportunity to talk about this biomarker-based dosing strategy with physiologic closed-loop controlled spinal cord stimulation. As some of you may know, ASPN has recently written a guidance that was just published on guidelines surrounding physiologic controlled spinal cord stimulation therapies and devices. And how did we get here? Well, I think we got here largely because with fixed output systems and not having the ability to measure the neural response of applying electricity to the spinal cord, largely what we've had is this black box of, are we activating, are we not? We had activation confidence when we used paresthesia-based systems. And with paresthesia-based systems, largely we solved that problem by using waveforms that don't require paresthesia in these paresthesia-free modalities. And with those, the same challenges arise where we don't know how much neural activation is required for a therapeutic response. And so largely what we've done is we've changed waveforms, we've changed targets, we've changed cycling strategies. And then with that comes this murky water of electing or deciding what to do when we're applying electricity and we're not getting the therapeutic response that we look for. So oftentimes in clinical practice with fixed output systems, we would trial a reprogramming, see if that helps. We will get x-rays, make sure the device is in the right spot. But largely, those are all largely surrogates of, are we neural activating? And then as a secondary strategy, if we are neural activating, is it therapeutic? Do people get benefits? So if you want to go to the next slide, Michelle. So what are the standards associated with spinal cord stimulation today as it exists? So we know that typically we do a trial. Usually that trial is either 3 to 5 or 7 days long. Some of us assess these outcomes of patients, usually as defined as a 50% pain improvement with a functional improvement throughout the trial, or some of us do so where we put the wire in and then we see people when we're getting ready to pull the wire 7 days later. And we had a Sonoma spine event last year, and it was interesting when we had a panel. Everyone does trials differently, so there's heterogeneity with that. We know that once we put the stimulation therapy in, because with fixed output systems, it doesn't really accommodate the resistance or the impedance associated with someone being living and breathing and moving. And with positionality changes, it oftentimes requires optimization. Data here suggests 6 times a year or more. And then when we think about folks that require optimization, and we think about days where they're not getting the significant improvement, we almost have a 1/6 of a year associated with people that have suboptimal pain relief. And then folks always like to, I think, point towards the fact that I published some data looking at failure of therapy, which was also echoed over the pond by another researcher. And largely, we know that devices fail as represented as a removal of the therapy with upwards of 11 to 22% within 2 years. So there obviously is room for improvement. And I think the question becomes, what could that be? Not only are our patients seeing maybe the need for optimization of delivering electricity to the spinal cord, but we also see other people paying attention to this. So we've heard of the JAMA article. We know about the Cochrane review for low back pain. We look at some of the payers that are starting to scrutinize spinal cord stimulation. Clinical effectiveness has been questioned or evaluated. And we have a lot of therapies or a lot of studies that are essentially asking the simple question, does spinal cord stimulation work? And I think for those of us that are on the call and those of us that have used spinal cord stimulation, I think we have a good sense that it works. But clearly, I think there is the ability to do it better. And I think what wasn't highlighted here is recently there was an insurance determination surrounding the length of trial, the testing that's required to do it. And again, that may be focused on answering the wrong question. And I think the question is, can you detect neural activation? Can you do so consistently? Can you titrate from success instead of away from failure? And I think hopefully that's what we'll see on the presentation tonight. So Michelle, if you want to go to the next slide. So Dr. Deere alluded to this before. There is a randomized prospective multi-center self-selected crossover study looking at spinal cord stimulation with a fixed output therapy as compared to a physiologic closed loop controlled therapy. And I think when we think about what that entails, at its essence, I think it is biomarker-based dosing. I think it is individualizing spinal cord stimulation to the patient, appreciating that everyone's got a sensitivity that's different. And there's been publications that echo that sentiment. So I commented on this 36-month randomized controlled trial, the EVOKE study. I think we're all familiar with that. There's been biomarker guidances that were recently published. And then the question that arose was, how soon can we detect a signal of neural activation that's therapeutic? And we recently published on predictability of trials. So if you want to move to the next slide, Michelle. So 30,000 foot view, when you look at the double-blinded, multi-center, self-selected crossover study, we see these things that I think we've seen that's been published across our space. You know, we have 80% of people getting at least 50% relief. We have 60% of people getting 80% relief or more. I think what's really telling about this technology is the fact that for those people that got success at 3 months, there is greater than a 90% predictability that they would also do well at 3 years. So we're getting really to the heart of, does spinal cord stimulation work for the long term? With a 36-month outcome in a prospective cohort, which is the first time we've ever had data out that far, I think the answer is yes. We can develop a spinal cord stimulation technology with a predictable outcome and longevity associated with the therapy. And for those that were treated in a double-blinded fashion, not knowing what they were getting or where they were going during the crossover design at 24 months, what we see is that over nearly 90% of people selected the controlled loop therapy or the physiologic closed loop controlled therapy. And of those that cross over from the control arm, which was the fixed output, they wanted to see how life was like on the other side, not knowing what they were getting and where they were going. And that was from the physiologic closed loop controlled side. From the fixed output side, they essentially wanted to have better pain relief. And of those that experienced the crossover, 90% of people chose physiologic closed loop. So I think that's really telling. I think the power in that study is not only the longevity, but also the crossover. Interestingly enough, if we talk about this concept of knowing the dose and controlling the dose, which Dr. Anthony is going to talk about in a little bit, once you get it dialed in, the question is, how often do you have to mess with it? And we can see that following the people through that cohort, that once you get it dialed in, it works pretty well because it is automatically adjusting the output of the device to maintain a neural activation 4 million times a day. So the consistency is there, which is evidenced by number of button hits and how often do they need a reprogramming. If you want to go to the next slide. And then again, this concept of, do trials actually predict long-term success with spinal cord stimulation? This has always been kind of a challenge because with fixed output systems and even with physiologic controlled systems, the trial is the best thing we have. And we're really dependent on a patient-reported outcome of at least 50% relief to tell us whether the therapy is working or not, without really having visibility on therapeutic neural activation. So with the new technology, we essentially did a study looking at the ECAP study cohort, looking at how early can you detect a trial success, and we define this as a day 0 trial. And what's represented in this would suggest that you have a 98.4% positive predictive value of determining success in a trial at day 0 versus day 7. When you have pain intensity reduction of at least 50%, you have functional improvement, you have an ECAP that is stable and durable. So I can tell you with a lot of confidence that during the trial, using physiologic closed loop controlled spinal cord stimulation, it does offer predictability associated with a 7-day. And then that's a chapter in the book of day 0 to day 7, day 7 to 12-month outcome, which we're getting ready to publish, but we have data that Dr. McHale already spoke on from 3 months to 36 months. So predictability is really critical. And my father's a PhD, my brother's a PhD, so he talks about not only do you have to have an outcome in a study, but you have to be able to replicate it. And so we see here across the publications that are on this slide, this has been validated across the pond. This physiologic closed loop controlled strategy has been validated within studies in the United States. It's been validated with a real world cohort, and there's continuing innovation that continues to occur, which includes the neural panel study and the Rosella study. If you want to move to the next slide. And how does this all work? This is my setup for our next speaker. We get these outcomes, we get the predictability, we get the durability by knowing the dose of neural activation, and that is for a therapeutic benefit, and then controlling that dose accurately and maintaining it throughout someone's life, whether they're sitting, standing, breathing, walking, whatever else. So we're knowing the dose and we control the dose. And that is in essence what a physiologic closed loop controlled device does, and specific for spinal cord stimulation. That's why I think we're getting the predictability and the outcomes and the durability that we're seeing with this therapy. So I'll pass it back over to Dr. Deer.
Jason, thank you so much. I think David's joined as well. Fantastic discussion. 2 quick questions. One is for me, you mentioned outcomes, and we're really blessed to have a lot of good therapies currently available out there, and many times we have great success. But you were with me in practice when you wrote that classic paper on failure. Do you see a role of using this data, this objective data, to lower the failure rate and also to maybe salvage devices that might be failing?
Great question. So if we're talking about outcomes, I think we're really focused on a pain intensity reduction as defining success with spinal cord stimulation. I know that Rob Levy and INS put a pain impact guidelines together looking at functional improvements across multiple domains like sleep and functionality, pain intensity reduction, mood. And so I think that's going to be the future in how we assess success with these therapies. Can we salvage someone with a fixed output system using a biomarker-based strategy? I think the answer is yes. So I've seen folks that have had therapies that have failed in the past that we've had success with this therapy, but I think the data is lacking, and I think we need more information.
Awesome. Jason, I have one question for you real quick. You've done a lot of work again on plants and when devices fail. Just a quick one. In your research, is it mostly poor patient selection, or is it the device or the waveform or something like that that leads to the majority of these failures? And can we use closed loop stimulation to to solve that issue if it's really more of a device thing?
Love that question. I think that I can tell you with confidence that the reason people got their devices removed most commonly was related to lack of therapeutic benefit. So whether that therapeutic benefit was related to device or whether it was related to patient selection, one thing that you can lean on biomarker-based dosing to do is to tell you whether you're actually neural activating therapeutically or not. So I'll back up a step. You can use it to definitely know whether you're neural activating. So the question becomes, is that neural activation therapeutic? We've never had that before in our space. We've never been able to say, what's the therapeutic dose of spinal cord stimulation, because it's been a black box. So I think with biomarker-based physiologic closed loop controlled therapy, the likelihood of poor selection goes down because you can neural activate, determine therapeutic benefit, and then move forward because the dosing paradigm that you're getting during the trial is the same that you would be getting at 36 months. So it's consistent across the board.
Jason, I just want to say that that last study you presented that you led on trialing is day zero prediction. I think may change the space. You get away from trialing down the road. Great job. Thank you for such a great presentation. We'll if we have time at the end, we'll come back for some more questions. I think Dalwa's going to introduce our next speaker now. Thank you
That's
so
perfect.
much.
Thanks, guys.
Thanks, Jason. So our next speaker is Dr. Anthony, a great friend of ours and a leading researcher, leading edge of innovation in spinal cord stimulation and neuromodulation now. What, you've been around 10 years or so now, I think. It's the times are catching up to you as well. You're not the young guy on the block anymore, Dr. Anthony, but he's going to talk a little bit about, knowing the dose and now controlling the dose. Dr. Anthony, take it away.
Thank you so much, Dawood, and you're dating yourself a little bit. It's really an honor to talk about this. This is something that's been a part of my practice and also a great area of interest in our research as well, collaboratively with a lot of people on the call here. Spinal cord stimulation is a large part of many of our practices, and I know that there's varying degrees of experience and knowledge related to closed loop stimulation among those observing today on the call. Many of us have probably utilized this in our practice, and some of us are just learning what this is about. And so let's talk about exactly what's happening with dorsal column stimulation, and then we'll move forward into talking about what's actually happening with the closed loop sensing technology. Some of those questions that were being discussed at the end with Jason and Tim and Dawood are questions that are going to help shape the future of our field. But before moving forward and talking about ECAP sensing technology and the closed loop system today, let's talk about what's happening at the physiologic level with dorsal column stimulation in general. So as we know, the lead is in a relatively fixed position in the posterior epidural space in either the cervical or the thoracic spine, but the spinal cord and the spinal column is dynamic. We move, we lie down, we breathe, we stand up. And as we do these normal daily activities, that lead, which is in a fixed position with fixed output systems, is delivering varying degrees of current or electricity to the dorsal column, which is moving in this column of fluid. And so dosing of spinal cord stimulation or current delivery to the dorsal column has been largely variable or a black box because of its variability with the dynamic nature of our bodies during life. With fixed output systems, the dose that's being delivered to the dorsal column is variable. Let's move to the next slide and we'll talk about kind of how we are trying to overcome that and understand how to be more objective with how we are maintaining the proper dosing of the spinal cord. And when we talk about dosing, we're talking about current delivery at the appropriate level and maintaining that through the life of the implanted patient. And as Jason was discussing, this is what we feel is potentially our best strategy for not only efficacy, but durability, maintaining those outcomes published out to 36 months. And hopefully we can look at outcomes even farther beyond that. When we talk about fixed output systems, we'll refer to this term fixed output as spinal cord stimulation in the history of spinal cord stimulation since FDA approval in the 1980s, all the way up to commercialization of the closed loop system or the smart loop system, which we'll talk about today. Fixed output systems, meaning we set the parameters of energy delivery, manipulating several things, whether it's basic parameters such as frequency, amplitude, and pulse width, or even more advanced programming options that you may see on the screen, such as waveform manipulation, frequencies, which can be high frequencies, low frequencies, novel energy delivery patterns such as burst stimulation, clustered tonic stimulation. Whatever you want to do with the energy delivery has in the past been fixed, meaning we set it, and that energy delivery parameters are not going to change unless we manipulate it with a remote control or programming that's done in the clinic. With a fixed output system, we don't have objective data or an objective view of the amount of neural activation that's happening in the dorsal column, and therefore we don't know the quote-unquote neural dose. And so what happens with uncontrolled neural dosing, which you see on the right side of the screen, at times the patient may be receiving increased amounts of energy delivery, leading to variable amounts of neural activation or evoked compound action potentials in the dorsal column. And at times, with movement, with lying down, there may be more energy delivery. With standing, walking, more activity in a non-supine position, there may be less energy delivery. And so that can be variable over the course of time, leading to potentially loss of efficacy, potential a reason for explant, and overall a potential reason for device failure. As Dr. Pope mentioned, both in Europe and the United States, under his lead and also Dr. Van Boyten's lead, the number one reason that we can say confidently in large studies is a loss of therapeutic effect. And so the goal of this stimulation idea is to maintain the optimal neural dose for durable therapeutic effect and prolonged efficacy from the device. Jason also mentioned the life of the implanted patient could potentially be different. When we talk about reprogramming burden, and that burden can refer to burden of the patient having to come to the office multiple times a year for reprogramming of the device to try and optimize the efficacy of the device, to even a burden on the practice for having to accommodate clinic space for patients to come, as opposed to potentially seeing new patients, talking about clinic workflow. There is a burden to high frequencies of reprogramming a spinal cord stimulator. And with a closed loop device in which we are maintaining a steady neural activation at a prescribed ECAP dose, the need for reprogramming is potentially less, and that's been published and also seen in clinical practice. Next slide, please. So this is an ECAP. For those of you who have had experience with the closed loop system, this is something that you can see on a clinician programmer. You can see as current increases, the morphology of an ECAP develops, and we are looking for a nice evoked compound action potential that you see in the middle of the screen here. What we want to do is achieve a nice morphology of this ECAP and also receive an ECAP measurement that's given in microvolts, and that's a number. It's not necessarily a window, it's a therapeutic target. That therapeutic target is then designed to be maintained for not only the trial, but for the implanted patient at the optimal level of ECAP activation that can be achieved at variable amplitudes of current, which we'll see in a few slides here. But thinking about fixed output systems, what you see on the left and what you see on the right can be happening at any time, and we don't have a look into what's actually happening at the neural level with a fixed output system. Many times a patient may be running the system at a subtherapeutic dose or subtherapeutic neural activation, which you may see on the left side of the screen as you see a very small ECAP starting to develop. Subtherapeutic dosing of spinal cord stimulation is a well-known reason for failure. The system is not optimized, the current is not high enough to achieve the level of dorsal column activation needed for pain relief, and subtherapeutic spinal cord stimulation energy delivery is going to be a reason why that patient fails. That can be perceived in several ways. If you are using a paresthesia-free system, then regardless of adjustments made on the device, there always could be a subtherapeutic level of neural activation, even if the patient perceives that it is helping some. Perhaps it's not helping as much as it could with the optimal neural dose or ECAP activation level. On the right side of the screen is, I think, something that we've all seen in clinical practice and heard from a patient in which they got temporary bursts of supratherapeutic stimulation or perhaps strong paresthesia from the device. And it's natural human instinct. I've heard it from my patients, time and time again, that with a paresthesia-based system, patients move a certain way, lay down in a certain position, and get strong paresthesia from the device, leading to a tendency to turn down the system. And that fear or that desire not to receive that strong paresthesia from the device can then lead to prolonged subtherapeutic neural activation, and again, poor efficacy or not the optimal efficacy from the device. So all kinds of variables that you see here that would not lead to the optimal neural activation that we see in the middle here with the good ECAP set that we see at the prescribed neural dose. Next slide, please. So how do we control the dose? What the system actually does is measure evoked compound action potentials from the dorsal column at the level of the electrodes and adjust the current on a real-time basis with every pulse. Next slide, please. This adjustment every pulse is very, very important for consistent neural activation. And so I think if we move to the next slide, we can see a picture of what's actually happening at the electrode level. The system evokes an evoked compound action potential by delivering the pulse, stimulating the target nerves in the dorsal column. It senses the evoked compound action potential, and again, that's not a window of a neural activation. It's a target level or target number given in microvolts, and adjusts the system on every pulse. This maintenance of the prescribed neural dose leads to no overstimulation, no understimulation, but consistent optimal stimulation for the patient's pain relief. And I think that we can think about consistent neural activation or consistent ECAP amplitudes at the prescribed neural dose as being synonymous with consistent pain relief. And that's what we've seen in the 36-month data. Next slide, please. And so this is a neural activation plot. I think this is one of the key differentiators of this system in thinking about what's unique in terms of consistent neural activation. This neural activation plot shows ECAP amplitude as a function of current. And as you can see, as the current on the x-axis increases, the ECAP amplitude starts to rise at this nice neck or elbow of the ECAP that we see. And from this ECAP activation plot or neural activation plot, we can set a target dose or the optimal dose for the patient. You can see that the target dose is above the ECAP threshold, but below uncomfortable paresthesia or the maximum that can be set with the device. And so patients do have the ability to change the target dose, but we know that we want to keep the patient with consistent neural activation and avoiding uncomfortable paresthesia or above supratherapeutic stimulation levels, which would produce too much neural activation. Next slide, please. So this is an individualized therapy. This is a therapy that is unique to each individual patient. You can think about it as each individual patient having a neural signature, and that's going to look very different for different patients. What we see here is two different activation plots. And you can see that the slope of these activation plots vary greatly. And we refer to the slope of these activation plots as sensitivity. You can see on the left, a patient with quote unquote low sensitivity, incremental increases in amplitude produce very small changes or increases in the neural activation or the ECAP levels in terms of the microvolt number that's being reported back to us by the system at the electrode level. What you see on the right side of the screen is a patient with very high sensitivity, meaning very small doses or very small increases of the current can produce very large changes in the neural activation or ECAP amplitudes. And we can see this on each individual patient, determine their sensitivity, and determine what their proper neural dose is. What I would ask you to think about is what happens for patients who we cannot see this information from. We do not know their sensitivity because we don't have these objective neural measurements. You can see for a patient with high sensitivity, it may be very, very difficult to keep them in that therapeutic window or therapeutic target to avoid over or under stimulation without this objective neural data. And what we know is that different, physiologic conditions or pathophysiologies, such as CRPS, may have high sensitivities. Different medications can influence sensitivities. We're just at the tip of the iceberg to understand, what influences these variables for each individual patient. So there's lots more to come. This is really kind of the beginning of what I feel is probably going to be the standard of care at some point. So we can move on, and I think we're pretty much finished with this section. Again, fixed output versus smart loop technology, different things. You can see on the left side of the screen, variable ECAP activation with a fixed current. On the right side of the screen, we see consistent neural activation, the system adjusting the current with each pulse and maintaining a steady level of neural activation. So very, very interesting and exciting work, and I'm happy to be a part of the research and also being able to help my patients with this therapy.
Really great job, Dr. Anthony. I just want to ask you a quick question. We've talked a lot about research and all the kind of amazing physiological data we can pull, but you're a busy clinician. You were smarter than me. You left academics and went to private practice. How has this been? You've treated a lot of patients with, I don't know, hundreds or whatever, How has this been to implement into your practice? We all heard the stories when this first came out in the clinical trials. It takes forever to program. You have to put the leads in this kind of, you got to go back to, doing paresthesia testing. And we had kind of simplified SCS for a while there with anatomical placement and not really having to on-table test them. How has this been to implement into your practice as you're a very busy private practice doctor?
Yeah, so I think it's made things a little bit more consistent in terms of what we can expect. So, during the trial and then certainly for the implant, we're able to quickly, know that the device is doing what it's supposed to do. I will certainly say that, for poorly selected candidates for neuromodulation in general, this is not a fix-all for every sort of problem related to treating a patient with chronic pain. But for a good candidate for neuromodulation, we are able to know the device is doing what it's supposed to do. And if that patient comes with pain, we can quickly know that the device is optimized and certainly treat other problems or address whatever the system may be. With fixed output systems, there are more variables that we have to tease out, as you once taught me, in terms of, troubleshooting a patient who has a problem with pain with an implanted spinal cord stimulator.
Yeah, I agree. Kind of the same, and I love that consistency. Tim?
No, Jay, I have a question for you. So, I've had 3 different groups of doctors text me while we've been on this webinar, watching this together. 1 group in Australia and 2 groups in the US. And I think obviously the question for a new doctor looking at this type of therapy, you said it was at 40 times a second, I believe you said, but it was actually measuring and responding. What would they ask you have a clinical specialist or rep in the room programming your patient, you're new to this, you see them in recovery, you see that response that Jason talked about, you see them a few weeks later after a permanent, we see what you showed a moment ago in the therapeutic window. Just give us a 30-second basic, what are we looking for as a physician to help us understand what we're seeing here?
Yeah, so you're gonna look at an activation plot, and that is kind of the bread and butter of understanding the system. You are going to get an ECAP amplitude. You're gonna see the ECAP developing as current increases. You're going to set a neural target, and that is going to be a number. It's not going to be a therapeutic range where you're kind of playing within the bumper cars. It's going to be a therapeutic target. You're going to make sure that the device is adjusting the amplitude to maintain that therapeutic target, certainly asking the patient to do different positions, to cough, to stand up, to lie down, and making sure that the perception to the patient is consistent, as well as the neural target dose remains consistent with those variables. And once you do that, you've established that the device is doing everything that it's supposed to be doing. the patient has confidence, the clinician has confidence, and quickly over the first weeks to several months after the implanted patient has this optimization process, as the tissues heal and kind of the signal stabilizes, that remains consistent throughout. And then we kind of are in a set it and forget it mode where there's not many interactions with the patient with the remote, reprogramming visits are less, and we kind of have consistency and confidence that the device is optimized.
Great, thank you so much. A wonderful job. I think you really taught myself and everyone listening a lot, and maybe we'll have some time at the end for some more questions. So thank you very much, Ajay, and thank you again. So our next speaker, our last speaker of the webinar, and then we'll also be taking some more questions at the end. Biomarker-based dosing, we've been talking about that. We've been building that with Jason and Ajay giving you the background. Now, Dr. Erica Peterson, I've known for many years, and her level 1 research has been outstanding. Her work in many areas of our field, but her work particularly here has been very impressive. So Dr. Peterson, I'll give you the podium.
Thank you, Dr. Deere, and thank you for that last question to Dr. Anthony, because the question of how do you use what we've just been talking about with understanding the specific individual information for patients, how does that translate into what we do in clinical practice is really the fundamental way of how we integrate this. So we've already heard about knowing the dose, this idea that there's a very customized response curve, and some people are very sensitive, as you saw, and other people may be less sensitive. So there's a customized, individualized dose curve for each patient. So once you know where that individualized dosing is based on what the threshold of when you first detect an ECAP, then we can set what that targeted dose is, how much stimulation current is required in order to generate the ECAP that suggests effective stimulation. And then how do we know that we're maintaining that dose along the way and have a nice conformal amount with a deviation from that target over time? So both knowing the dose and controlling the dose then translate into how do we optimize this for each patient, as well as how do we maintain that effective stimulation over time so that we don't have loss of efficacy, as Dr. Anthony already talked about. Next slide, please. So there's 2 papers that are follow-on that I've been honored to have a contribution on that really look at applying these findings to the actual practical portion of it. And this is where we look at actually how do you use the biomarker of the ECAP and neural activation to dose for patients. The first of these looked at 690 patients out of several of the studies that Dr. Pope listed for us initially, talking about what are the things between dose accuracy and variability that ultimately lead to successful results in long-term durability of outcomes for patients. The other paper looks at maximal analgesic effect, and we looked at a subset of 180 patients who were within these studies, and what we found was that there was a point when patients had their maximal pain relief, their maximal analgesic effect. And we could correlate that to what was their ECAP dose, what was their therapy set at the time of that dosing. And so what we're getting to is a point where we can actually know by patient a guidance on how to set the dose of stimulation in order to have the best chance of getting that maximal analgesic effect. So we have other things that we've needed to titrate in terms of medications, and now we're talking about titrating and individualizing dosing for neurostimulation. Next slide, please. So this is now thinking about things in terms of prescription. So if you know what the person's ECAP dose is, are we optimized? Does that line up with the research that I just showed you in terms of where the maximal analgesic effect was for the patients who've been studied? And then can we look at dose accuracy? How much variability is there around that target in terms of actually holding conformality and getting that as close to that target dose as possible over time? So again, each person will have their own dose curve, and that's really important. I'd like to suggest that if you think about the analogy here, it's not just like medication, but think about, when I go to a bar, I know I'm a beer and a half kind of person. That's going to get me to my happy spot, but somebody else may be a 5 beer tolerance type person. And so we have different dose responses and dose ratios. And that's the same sort of thing here. We need to be able to individualize this to patients. And the key to that is being able to absorb and see what that that neural activation is as recorded through the ECAP. Next slide, please. So what did we actually see with that? Well, so when you look at where people are, what we find is that there seems to be a ratio that people find their maximal analgesic effect somewhere between 1.2 to 1.6 times the ECAP threshold. That is not current again, but the current that it takes to get to that place on the curve where you are 1.4 times the ECAP threshold. And so once you're at that location, can you hold that by delivering variable current if necessary as the patient is dynamically active through their day with coughing or breathing and twisting and bending and all the other activities that they go on. And that's where the 40 hertz, 40 adjustments per second matters, because with every impulse that is delivered, there is the sensed ECAP return and then the adjustment of stimulation that's provided in order to try to continue to maintain that targeted optimal ECAP dose. Then when you control that dose, by doing that, you can look at the variability. Next slide. So I want to give you an example, and this goes back to what Dr. Anthony and Dr. Deere were talking about in terms of what do you do in clinic. So here's a patient who has an implanted closed loop stimulator using the ECAP technology with Saluda. This patient at the 1 month visit says that they are having 81% pain relief. And when you look at the data that's downloaded from the stimulator in terms of their device performance, what you can see is that their therapy dose ratio is 1.30 times the ECAP threshold. So it's right in that range I just showed you along the curve. And their dose accuracy is plus or minus 7 microvolts. So they're pretty conformal along that variability around the target as well. But when they come back 3 month visit after implant, they tell you, you know, doc, I don't like this. It's not working. I've only got 45% pain relief. This thing is a piece of junk. If you didn't have the objective data of the neural panel where we could actually download the statistics, you would probably say, let's reprogram, let's maybe get some x-rays and see if the leads had moved. Let's try to readjust where you are with stimulation. Can we try a different waveform? You'd start to troubleshoot, and that might be an iterative process of several visits with the reps in the office, each one taking time from the patient, taking time from the rep and from our office staff in order to try to optimize. But in this instance, what we can do is review the device data, and what we can see is that the device data shows that at some point the patient had turned down their stimulation, and now they're only getting 0.61 times the ECAP threshold. They are getting a sub-threshold amount of stimulation, which is nowhere near that target of 1.3 to 1.4 times the threshold. It's half of that. So does it make sense that they wouldn't be getting maximal analgesic effect when they're nowhere near that target? They have dose accuracy that's around that, but their target is completely out of range. So because you have that objective data on the patient, we know exactly what needs to be fixed in order to optimize the stimulation back into a range where we can be sure that it's delivering technically excellent stimulation. Then the patient comes back, and at the 6 month visit, they're reporting 94% pain reduction. They've been doing well, holding on to the stimulation guidelines the way we set them. Their clinical outcomes now are maintained. They may even be able to decrease some of their medications because of the improvement that they've had. And what you see at those is that when you download the data, they're at 1.2 times the ECAP threshold. And again, they have very conformal dose accuracy with that plus or minus 7 microvolts. So even if the dose accuracy is great, if you're not giving it something at the right therapeutic ratio, you're not going to get the benefit. But then again, you may also have a patient who is at the right dose ratio, but that right column with dose accuracy may be a much higher number, which means there's a lot of fluctuations. And if you have fluctuations and you don't have that conformality around the targeted dose, then the patient may not have the kind of optimal result we're looking for either. So those are really important data points that help me make a decision. I feel much more comfortable doing data-driven decision-making the same way I do looking at lab results, for example, than I do just going off of subjective information and trying to make best guesses on things. And so a data-driven way of trying to help inform patient therapy is a really new innovation in a way that I think helps to improve our practice. Next slide, please. So here's another example. So in this case, the patient came in, and this is exactly what I was talking about a second ago, 66% pain relief, but you can see the dose accuracy is plus or minus 43. Their therapy dose ratio is really good, 1.5. So can you get that more conformal? Absolutely. Let's bring that ratio around the dose accuracy down to plus or minus 9 microvolts. And now they're a little bit better, 68% at the 3-month visit, but you give them a little bit more time. And at the 6-month visit, here they are with 89% relief, and you can see again both their therapy dose ratio is 1.33 times the ECAP threshold, and their dose accuracy is plus or minus 7 microvolts at that. So again, coming out of an out of range, less conformal towards a more optimal stimulation range based on the data that's come from the level 1 evidence that we've pulled together from studies looking at closed loop outcomes, which includes the patients with 36-month following of their benefit, has really shown a way that we can try to optimize this for patients in the clinic. Next slide, please. One of the things that I think is I really want to highlight is that this has been something that we've seen over several different studies. So each one of these studies on the curve, you can see there's a slightly different dose ratio range between the different studies, but all of them are in that 1.3 to 1.4, maybe a little high at 1.6 dose ratio range. And that's why we say that 1.4 is sort of the place to be. It is what the current is in order to get that ECAP dose compared to the ECAP threshold, and it's then really in that 1.4 range that we think is going to achieve the maximal analgesic effect for patients. Now, what I need to know in order to dose a patient is, what does that patient's individual curve look like? Where is their ECAP threshold? Are they a hypersensitive patient? Are they less sensitive? And once I know what the ECAP threshold is and what the current is to achieve that, I know that I can get to that 1.4 times the current up along the curve in order to set their initial dose for therapy. Now I'm not going to be guessing and trying to sneak up on it and, oh, try another click, or let's try this other thing, but that we have something that it's actually much more prescribed and much less vague in terms of how to direct using this therapy for our patients. Next slide. So one of the things that I think is really important for all of this is the objectivity of these data. So I've touched on the fact that this is something that I think is really fundamental. You have this idea of having the objective neural activation. You can see what the ECAP plot looks like. You can see where the activation is for each patient, and you can see the conformality of every piece of stimulation that's delivered to that patient for every impulse. And so this kind of objective data is really groundbreaking. It's the sort of thing that I think is going to help me drive my practice, and already is driving my practice, and I hope will drive other practices to help find ways that we can optimize stimulation for those patients who may otherwise be considered stimulation failures for loss of efficacy, because now we actually understand what is happening within the spinal cord in terms of how we are actually seeing neural activation and changes related to the neurostimulation we're delivering. Next slide. So finally, I just want to wrap up and say that you've heard from 3 of us tonight talking about the power of these data and how we've taken and applied this to each one of our practices in different ways. It's a really powerful opportunity. It's certainly not the end of what closed loop is going to be. I'm really optimistic for this as the start of a closed loop journey on things, but I really feel the power of the data is important, and I hope that I've shown some good examples of how that biomarker-based dosing can really lead to predictability and dosing for individual patients to help guide those decisions in what may otherwise be complicated cases.
Oh, fantastic job as always. Certainly, that's really a new groundbreaking discussion that I really haven't heard before. A couple questions for you. I think we're gonna have 2 each as we finish up on time here at the 1 hour mark. My first question is: you have a patient who is morbidly obese, diabetic, drinks alcohol every night, isn't very healthy. You put this device in, you measure the biomarker window we just pointed to. And it's a 2-part question. One is, if that person gets healthy, controls their sugar, moderates their alcohol use, will that biomarker change? Are they a different person? And what's the future? Will it be artificial intelligence automated programming in the near future, do you think?
Yeah, so there's really good questions about what happens with ECAPs, and we've had some initial research that shows, for example, that as medications change, or even as somebody becomes dehydrated, that where that ECAP threshold is may slightly change over time. And so, so I think it's very important that we are aware of that, particularly when patients are having medication tapers or other significant health changes, that we reassess and we can create an activation plot at any time. That's something on any visit that a patient's in the office, the first thing you do is say, let's build an activation plot and see where that ECAP threshold is. And then again, that ratio that's been shown over 700 patients comes from all sorts of patients who have lots of different comorbidities and leads at different target locations as well. And so it's very interesting to see that that ratio somewhere in the 1.2 to 1.4 times the ECAP threshold is consistent, even when that happens. So if somebody is showing a change in loss of in terms of their therapy benefit, redo the curve for them, and then reset and make sure that you're really back to that 1.4 times threshold.
Great job, Eric. I have a question for you too as well. So as a neurosurgeon who deals with, neuromodulation devices, just even outside of just chronic pain, like those of us that are kind of devoted to just chronic pain management, closed loop technology, there's another one already out on the market. I think others are working on them. It just makes so much sense. It seems like it's such an easy concept for me to explain to my patients. They're like, oh, why haven't they done this forever? What do you see with closed loop technology, outside of the realm of just chronic pain? Do you see it having a bigger role in movement disorders, deep brain stimulation, et cetera?
Well, I hate to break it to you, Dawood, but closed loop's been around for cranial stuff for like a decade. And so the responsive neurostimulation devices that we use for epilepsy are designed to sense pre-epileptiform discharges and then deliver pulses to stop the kindling of a seizure. And so those have been around to be continuously sensing and then responding pretty well. The deep brain stimulation devices for Parkinson's are designed to record the local field potentials within the brain and look at beta band activity within the basal ganglia and use that beta band signal to then figure out how much dose needs to be delivered based on where we are in terms of dopaminergic deficiency. So that's been shown in dozens of patients and is starting to get more available. And so we're going to see more and more opportunities for that. I think one of the things that I'm most interested in is where can we go with spinal cord stimulation and closed loop spinal cord stimulation for non-pain indications, because the brain itself is something that is pretty invasive, and I certainly would prefer a non-brain procedure and a little bit lower risk profile. And if we could do something in the spinal canal that could improve a movement disorder or could improve spasticity from stroke, for example, or potentially the spasticity from CP, wouldn't that be an interesting direction to look? And so I think as we are able to control and understand neural activation, and we get away from the extraneous paresthesias and paresthesia mapping and focusing just on pain, there are going to be some really interesting opportunities for what we can do in terms of other things that are neurologically modulated in some sort of way. And the question is then, does that go into other non-neurological conditions? Where are we, for example, with potentially looking at metabolic disorders, which are a huge problem for people, particularly in the US?
Erica, I think, having Parkinson really becoming an epidemic in America, I think the gait disturbance part of that, we know for tremor, the brain is pretty successful, but the gait disturbance, I think, has great potential for closed loop. And I know you're doing some early feasibility work on that. And I think that'll be fantastic. We have 1 minute left in our time. So Dawood and I are going to close it out here. I will say, one thing you really explained well, I think the interaction with this rep locally, wherever you may be in the world, when you have those biomarkers to look at, it's going to really change the discussion when someone's not responding. You go back and you remeasure a biomarker. I think that really, your discussion really helps me think about how I may approach someone who needs to be, reprogrammed at some point if they have a change or something, because you can go back and say, is this the same as it used to be? And we can never do that before. So thank you so much for your time and your lecture. I think we all benefited greatly from it, as always. And we'll see you soon. Dawood, you want to say a few closing words?
Yeah, so as we kind of, did the Q&A throughout the program, we're kind of done with that part. Really appreciate everyone's time. I shout out to, we had, a couple hundred people or so kind of logged in, and then we had almost, I think up to 10 to 15 dinners across the United States in areas such as here in Kansas City, Florida, Baltimore, and even in Hawaii. And I think you mentioned getting some calls from some folks in Australia. So shout outs to all of you from around the globe who were able to tune in today. Thank you, everyone. We hope to do more of these in the future on this topic and many more. Make sure, you sign up for the annual meeting coming up in July 2025. We're going to have a really innovative agenda, and we'll continue to push the limits of what we can learn in this space together. Thanks, everyone.
Yeah, the annual meeting is July 17th to 20th, Fountainblue Hotel, Miami. It's on the website now if you want to sign up for the meeting and get your hotel space. By the way, I thought the day was great, Dawood. I thought the discussion was great. I think it was very objective and non-biased by the speakers. They did a great job showing the data. And I think we're going to see more and more companies come into the closed loop realm. And I think it's going to be the future in a lot of ways. And whether it be a ECAP, as this study was based on, or other biomarkers, I think biomarker medicine is the future of where we're going to go. And I think it will help us get better outcomes. So with that, everyone have a great night or day if you're here in my hemisphere. And we hope to see you all soon.
Thanks, everyone. Good night.